This video will cover subtopic 5.4, Cladistics. Here are a list of understandings, applications, and skills that we will cover in this video. We'll start by talking about homologous and analogous structures. We've discussed this in a previous video, but we'll go through this in a bit more detail again here, because this is a system of classification that is used. Analogous structures resemble each other in function, but differ in their fundamental structure. The example down here in the diagram shows the streamlined appendages found in sharks, penguins, and dolphins as an example of analogous structures. Now, they resemble each other in function, but they differ in their fundamental structure, and they really only demonstrate superficial resemblances. And these structures would have come about through similar selective pressures, for example, swimming through water. Now, homologous structures are built to a common plan, but adapted for different purposes. And the diagram down here shows the example of that pentadactyl limb. So homologous structures are similar in that fundamental structure, so they have that pentadactyl limb has similar bone structure. They are similar in position and development, but not necessarily in function, so that pentadactyl limb can serve different functions in different organisms, and are similar because of a common ancestry. Now, a classification system based on analogous structures is considered an artificial classification and does not show evolutionary relationships. A classification system based on similarities and differences, i.e. these homologous structures, is considered a natural classification system and can show evolutionary relationships. And in these classification systems, we are trying to show how things are related evolutionary-wise. Now, other characteristics that can be used for classification are the following. So the following table shows some types of characteristics that botanists and zoologists might study in order to help them decide how to classify an organism. So they could use morphology, anatomy, cytology, the structure of the cells or their function, phytochemistry, chromosome number, or molecular differences, so proteins and DNA sequences that can differ between one species and another. So looking at differences between DNA sequences or amino acid sequences between different species. And this is what we will focus in on for this form of classification. So looking at these characteristics used for classification, just a few things to recall: that all living organisms use DNA built from the same four bases, that A, T, C, and G, to store their genetic information. Also recall that most use the same triplet coding system during translation, that process of making a protein, of combining amino acids together. And this is found in all organisms except for mitochondria, chloroplasts, and a group of bacteria. So all living organisms are going to use that same triplet coding sequence. Now, remember that proteins are built up from amino acids, and all living organisms will make use of the same 20 amino acids. So in most cases, if a gene from one organism is transferred into another, it will produce the same polypeptide. Think about that example of gene transfer of insulin from a human cell to a bacterial cell. So these facts all indicate a common origin of life, and it suggests that all organisms have evolved from a common ancestor. The study of the genetic code and amino acids of an organism can provide evidence that links it to its close relatives. And this will allow us to build up diagrams called cladograms, which show how species are related to one another in what we'll call clades. And we'll go through examples of these and what these are in this video. So looking at clades and cladistics, so we'll start with cladograms. So cladograms are diagrams that will show species evolutionary relationship to one another. So cladograms, for our purposes with DNA evidence, take on this kind of tree diagram form. The branching points on cladograms, so you have these branch points here, are called nodes. So this point here would be considered a branch point, and this would be called a node. And nodes denotes speciation events where a common ancestor splits into two or more species. Now, the cladograms that we will mostly consider are based on DNA sequences or amino acid sequences in a protein. So it compares DNA sequences between organisms and then determines their evolutionary relationship in that way. Now, because these DNA sequences are very large, and amino acid sequences, we can have hundreds to thousands of amino acids in a particular protein. Software programs can be used to combine multiple sequences to find the most probable pattern of divergence or evolutionary relationship between organisms. Now, with cladograms can be drawn in one of two ways, so they can have two different shapes to them. So we have this typical tree diagram that we will consider mostly, and this type of cladogram is typically constructed from DNA or amino acid evidence. Now, the second type of cladogram that we have is constructed from comparisons of structural features. So in this particular cladogram here, we have the structural features that are being compared between these classes of animals or these particular groups of animals here. And the first divergence point, or the one at the bottom of the line that diverges away, shares the least number of characteristics with these other animals, or shares the least number of characteristics with mammals, for example, that is found at the top of this cladogram. Now, in cladograms, they can be organized into clades as well. So keep in mind that species can evolve over time and split to form new species. Now, consequently, there are groups of species derived from a common ancestor. These groups are referred to as clades. So species that share common ancestors can be grouped together in clades. Now, clades can be big or clades can be small. So in this particular example here that examines this particular cladogram, so we have species 1, 2, 3, 4, and 5 on this particular tree diagram. Now, as a whole, species 1, 2, 3, 4, and 5 all share an originating common ancestor, and so they would belong to one large clade. However, if you look at species 1 and 2, they share a common ancestor, and so species 1 and 2 would belong to their own clade. Now, if you look at species 3, 4, and 5, they also share a common ancestor, so species 3, 4, and 5 would belong to a clade of their own, and this is outlined in green. Species 4 and 5 share a common ancestor between them, and so they would have their own clade, so this is outlined in red. It would be incorrect to classify species 2, 3, 4, and 5 as one clade, as there is not a common ancestor between them. So 1, 2, 3, 4, and 5 all share a common ancestor, which is a big clade, but then you can have smaller clades within that big clade. Now, there are reasons for using cladistics to organize and discuss information. So it can be useful for creating systems of classification that can communicate ideas about species and the history of life, and it can be used to predict the properties of organisms, and it helps to explain and clarify mechanisms of evolution by looking at similarities between the DNA and proteins of different species. Now, the way in which we find evidence for clades and constructing cladograms is the following. So establishing clades and constructing cladograms is done using phylogenetics. And phylogenetics is the study of how closely related organisms are. Molecular phylogenetics examines sequences of DNA bases or amino acids in polypeptides of different species to establish the evolutionary history of a group of organisms. Species that are the most genetically similar are more likely to be closely related. So we'll first look at using DNA to construct a cladogram. First, I'll ask you to recall that when you have changes to the DNA base sequence, these are referred to as mutations. So when you start to change the letters in that DNA sequence, this is referred to as a mutation. Mutations in the DNA sequence of gametes are passed to offspring, and if the mutation is not harmful, it will be retained in the genome. So mutations only get passed to offspring if they occur within those gamete cells. Now, differences in DNA accumulate over time at an approximately even rate so that the number of differences between genomes can be used as an approximate evolutionary clock, which is referred to as the molecular clock. And we'll go into more detail about this molecular clock. And so what this molecular clock can do is it can indicate how far back in time species split from their common ancestor. Now, a greater number of differences indicates that there has been more time for DNA mutations to accumulate than if the number is smaller. So there is a positive correlation between the number of differences between two species and the time they evolved from a common ancestor. Now, looking at the molecular clock to help us create this picture, so these biochemical changes, these mutations, may occur at a constant rate and can be used as a molecular clock. A molecular clock will use the average rate at which a gene mutates to gauge the time of a divergence between species. Now keep in mind that mutations can happen at varying rates. There can be selective pressures applied to particular genes, and so it can affect the rate at which mutations occur. So it is important to keep in mind that with this molecular clock, it is an estimation and not an absolute time or date for speciation events. So it is an estimation, it's an approximation. Now, molecular clocks do have to be calibrated, and this biochemical data, these mutations, are compared with morphological fossil evidence and radioisotope dating to ensure accuracy of the timing. So that clock is calibrated relative to the fossil record and radioisotope dating. If the mutations or the changes in DNA match up with what the fossil evidence says, it indicates a close relationship. So this example shows a molecular clock with a rate of one mutation every 100 years. So here we have the current day organisms, and here we have the kind of common ancestor to them all, and the DNA sequence that may have existed in that common ancestor. And so between that common ancestor and the current species, there are a total of 4 different mutations in that DNA sequence. And so if those mutations occur, if you get about 1 mutation for every 100 years, it indicates that there is there's a difference of 400 years between these current day species and that common ancestor. And so if you look at this particular lineage here, the difference between this species here and that ancestral species is 2 mutations, which indicate that speciation would have occurred about 200 years ago. And then from this species to the current day species, there is an additional 2 mutations suggesting that the speciation event occurred 200 years ago. So a difference of 400 years between the current day species and species from that common ancestor species. Now, with the molecular clock, the strong linear relationship suggests that molecular differences between pairs of species are proportional to the time of their separation. Therefore, measures of genetic divergence can be used to date the time of divergence for species pairs for which no fossil data are available. So basically, if you have the here on our chart here, if you have nucleotide substitutions, the number of nucleotide substitutions that occur, and millions of years ago here on our x-axis, this is the time since the two species diverged. Basically, the longer the time ago, the more differences that you see in the DNA. So the greater the number of substitutions, the longer ago that these two species diverge. The smaller number of mutations indicate a relatively shorter period of time since speciation would have occurred. So molecular clocks also have to be calibrated, as I mentioned before, and they are calibrated using the fossil record or known times of geographic isolation. So this figure just shows the calibration of the alpha globin gene, and it basically shows the divergence based on the molecular clock. These are the red dots when you compare the number of differences between these different species. And then this is the divergence. The yellow dots represent the divergence based on the fossil record. So where in the fossil record you start to find the earliest ancestors of these particular species. And basically, you can see here that the closer that the two dots are to each other, the more accurate the estimates are of the date since the species diverged from humans. And so you basically see kind of the same trends and pattern here. So, the molecular clock does line up with that fossil record. Now, there are cladograms that exist for humans and other primates, and so the DNA evidence used to build this cladogram can also be used to estimate when species diverge, and hence when that common ancestor existed. Now, one of the things in cladograms, generally speaking, is that the length of lines separating species on cladograms is often used to represent the estimated time since they diverged. So the length of the line can show how long ago a species diverged. So in the following example, mitochondrial DNA from humans and primates has been completely sequenced and used to construct a cladogram between them. So that's what this diagram represents. And the rate at which these mutations occurred, these differences in the mitochondrial DNA, was used as a molecular clock to calculate how long ago these species diverged. If the DNA base sequences or two species are similar, then few mutations have occurred, and the species have only relatively diverged recently. So hence the shorter lines between chimpanzees and humans, as there are a high number of similarities between the mitochondrial DNA, suggesting they diverged very recently. Now, amino acids can also be used to construct cladograms. So related organisms will have these same molecules carrying out particular functions. These molecules will also have similar structures. By comparing proteins in different groups of organisms and checking them for similarities in amino acid sequences, it is possible to trace their ancestry. For example, chlorophyll, hemoglobin, and insulin have been used in this way. So here we'll look at an example of an amino acid used to construct a cladogram. So the beta chain of hemoglobin is built from a total of 146 amino acids and shows variation in the sequence of amino acids between different species. The more closely related species are, the more likely their amino acid sequence is to match. Or the longer ago a species diverged from a common ancestor, the more likely it is that these differences may have arisen. And this table here shows these differences, the number of differences in that amino acid sequence for the beta chain of hemoglobin. And so this is all compared back to humans. And so humans obviously have zero differences as a species. in that amino acid sequence, whereas gorillas have one difference between them. Gibbons have two, rhesus monkeys have four differences, or sorry, eight differences in that amino acid sequence, and so forth. And so the greater the number of differences that you see, the longer ago divergence occurred between those two species. So this could indicate that humans and gorillas are more closely related than humans are to frogs, for example. So divergence from frogs occurred longer ago than humans from gorillas. One of the things that you'll be expected to do is to be able to analyze cladograms to deduce evolutionary relationships. So the pattern of branching in a cladogram is assumed to match the evolutionary origins of each species. If two species are linked at a node, they are closely related. If two species are only connected through a series of nodes, they are less related. And so this diagram gives an indication of that. So here we have this ancestral lineage. This first branch point here represents a common ancestor to these organisms, to all these organisms here in this particular cladogram. And then this branch point here at 2 indicates where lineages have diverged, and then same at 3 where you get these particular speciation events. And this branch point at 5 indicates basically an unresolved pattern of divergence. So this is still being figured out, I guess, if you want. So in this particular cladogram, you could determine that the organisms in taxon B and C are more closely related to each other than to the species in taxon A. And the species in, for example, taxon B would be more closely related to the species in taxon A than the species in taxon D, as there are more nodes between the species in taxon B and the species in taxon D. Now, the following example has a series of questions that has you analyzing this particular cladogram. So, take a few minutes, answer the questions here. So, what animal is most closely related to a squirrel? What animals are more closely related to humans than our old world monkeys? Tarsiers share a common ancestor with which animals? Elephant shrews, elephants, manatees, and dugongs all share a common ancestor. Which animal diverged from the ancestor first? So reclassification has happened. The example that IB wants you to know about is the reclassification of the figwort family using evidence from cladistics. So the figwort family, the Scrophulariaceae was a large family of Angiospermophyta that included snapdragons, figworts, monkey flowers, and many other garden and wildflowers. Biologists had classified the Scrophulariaceae plants using external morphology features. This diagram indicates or shows the features that were used to classify these plants. So they all had these following features here. Now, a study done by Olmsted and al. In 2001 compared 3 chloroplast DNA genes, which totaled 4,200 bases across 65 plant species. The results were analyzed by a computer program to try and give the simplest explanation for the differences found. It was found that the species who were all originally in the Scrophulariaceae were not closely related to each other. As a result of the DNA evidence, the Scrophulariaceae family suffered a nasty divorce. The old Scrophulariaceae have now been split into at least 6 smaller clades, according to Olmsted et al. In the study done in 2001. Now, reclassification was done using that evidence of cladistics. So a number of plants remained in that figwort family. A number of plants got moved into a different family, the Lopseed family. A number of figworts were moved into the plantain family, and then other families, brand new families, were created, along with new clades were created as well. So the DNA evidence identified the different common ancestors, that the that the similarities between these plants were analogous. They were based on superficial structural features that looked similar, but were ultimately different in their underlying structure. That general flower shape and form of the seed capsule evolved many times from different ancestors, as an example of convergent evolution. So they had very similar selective pressures, and so these plants with different common ancestors or different ancestors all developed flowers that had a similar shape because of these similar selective pressures. And this was due to the fact that they were all competing for similar pollinators and all adopted similar seed dispersal strategies. Now, as new evidence is discovered, in this case DNA sequences, older ideas for classification needed to be reassessed and sometimes changed, and this is always an ongoing thing as technology gets better and we're now better able to study a wider array of DNA sequences from different organisms, new evidence emerges. So sometimes that new evidence shows that members of a group do not share a common ancestor, so the groups are split and moved into different clades. Sometimes members of different groups are found to be closely related, so the groups are merged into a single clade. Ultimately, the big takeaway from this reclassification of the figwort family is that biologists are constantly testing ideas and hypotheses. As new information is discovered, current theories or explanations may be found to be false. And so this requires new explanations and new theories to be developed. Therefore, scientific understanding is always changing and improving. It's never something that is static. It always has the potential to change as new information comes along. So scientific understanding is always evolving. So that is the end of this video for 5.4 Cladistics.